preparation. Okay, well, this is about stem cells and why they're a double-edged sword. And the reason why they are is the incompatibility, the immunorejection, because they're not compatible. So I have no conflict of interest. I'm a scientist. I'm not associated with any booths or anything. And just an introduction of what stem cells are. You have the totipotent, which basically can do just about everything. And you have the pluripotent that they can do extra embryonic tissues. And then you have the multipotent, which is cell types found in the tissue of origin, like the mesenchymal stem cells that we're going to be talking about later, and the unipotent, which is basically one specialized cell type. It's just the cell types. And the most important thing about stem cells is that the stem cells must differentiate. If they don't differentiate and they just proliferate, this is what cancer does. Basically, they're just going to sit there and create tumors eventually. So this is the most important thing to remember about stem cells. So cellular differentiation means forming a variety of different cells. So you can see here different cell types being kind of formed. And first is the embryonic stem cells. That's where it all started. And the application has been controversial because of ethical reasons, and also because they create teratoma cancers as a result of proliferation, but lack of differentiation. And basically, differentiation depends on mitochondrial activity. And the stem cells, as you can see, the disrupting mitochondrial function disrupts proliferation. This is a study that shows how when you disrupt the mitochondrial function, then you're going to disrupt the proliferation and the differentiation. And the mitochondrial activity increases differentiation, whereas the nuclear activity prevents differentiation. And the embryonic stem cells have a low ratio of that. Basically, they don't have enough mitochondrial differentiation because they don't have too many mitochondria inside. So that is the problem with stem cells. They have few mitochondria because they rely on anaerobic metabolism. So they don't have the same potential for differentiation, even though they can be translated in many, many different cell types, which makes them extremely kind of useful and popular. The problem with lack of differentiation, again, is going to be the uncontrolled proliferation, which will create, it's very similar because just like cancer, stem cells kind of, they keep multiplying. And this is, you see how a normal cell can have just one cancer cell that has been created by lack of differentiation, and how that can start increasing because the cancer cell is going to start multiplying as well. So just one cancer cell is going to start kind of increasing the teratomas, and this is what teratomas look like if you have an operation and you take it out. So again, the most important thing for stem cells is the immunorejection. And what does immunorejection mean? It means that the stem cells you've injected and your body are not compatible. They are not resonant with each other. And here I would like to introduce the concept of resonance, but in a very different way than the other speaker was talking about. So in both mouse and humans, even one mitochondrial mutation, just one mutation in the mitochondria can be enough to basically throw out the entire injection, because it will create an immune response that will create the immunorejection. So in order to avoid this immunorejection, there is a Chinese study. What they try to do is to genetically engineer liver stem cells that are invisible to the immune system. And they claimed that when basically the liver cells, when they could not basically camouflage the transplant to avoid the immunorejection. So it's not clear, because they said that if it's a tumorigenetic, if tumorigenesis begins, then the immune system will recognize it. So it's just sort of that you have it both ways. So a lot of people have been very critical and suspicious of this research. And then the Stanford University had a research where they used a pair of antibodies that blocked the normal T cell activation. And so basically to stop the immune system, to blind the immune system. But the problem with that is that, yes, you have the stem cells that can now kind of not be accepted by the system, but at the same time, the immune system is not working, so if something goes wrong with the stem cells, then you have many problems. So you basically win the battle, but you may lose the war. And then we have the Nobel Prize in Physiology and Medicine, that they basically found that there is a unidirectional process of stem cells, and that Nobel Prize basically created, reprogrammed cells to become iPS cells, which is basically can be differentiated in vitro into different stem cells, and that would be kind of something that can help. But the problem with that, and this is the different, I don't want to go too much into it because I don't have very much time. This is the proteins that they looked into the Nobel Prize. But the problem with that is that iPSC, or the stem cells, also can be highly mutagenic. And then you have the same problem of basically cancer formation. So the stem cells are wonderful until they're not, basically. So both the human-induced pluripotent stem cells and the nuclear transfer stem cells, they both have a potential for tumorigenesis. So an in vivo analysis of teratoma, this is some studies that I can show you how basically these stem cells created like that. And then we have the mesenchymal stem cells that are far more basically safer in that sense. The problem with those, with the mesenchymal stem cells, is that basically, and they can be used in a lot of medical cardiovascular diseases and brain injuries and so on and so forth. The problem is that from different donors, they can be heterogeneous, and the stem cell phenotype is affected by cultural conditions and cell passage. And also the most important problem is the senescence, that when they are senescent, they decrease their differentiation potential. And here you can see the early, under the microscope, a stem cell here, and then in the senescence, a completely different, it has changed. It's basically, now we have endogenous stem cells. Every time we exercise, the fat, as it goes into the bloodstream, it also releases stem cells. So you have endogenous stem cells. And there is a lot of research that is associated with that. And I can tell you, remember the other speaker was talking about intermittent fasting? Well, that can also, actually, I don't have it here, but that can also, I wrote an article about that, that can also actually have stem cells, create stem cells as a result of that. And this is some of the articles that I wrote about endogenous stem cells and how that they can help kind of repair the liver. And this is the results that you can see with effortless exercise. You see the everything's in the right direction. Basically, the ALT, the AST, the ALP were significantly reduced. The albumin was increased. And you had also creatinine and bilirubin, again, a decrease in you don't want to, the percentages might not be very high, but everything has to be within a normal range. You cannot just increase it kind of above the normal range. And then we did a longitudinal study, and you see that after 7 months, there was the same effects, that basically everything was in the right direction. And when they were like very low, then there was an increase. So the normal, everything was sort of normalized as a result of the exercise. So, and that's how, now, this is what I need to bring to you, how important it is, the systemic balance, because we do injections, but we have to keep in mind that there is a process of systemic balance. There is, the system has an interconnectivity, and this is something that we have to take into consideration as a very important thing. This is how cells work under the microscope. I have another lecture afterwards, you're going to see this again. But basically, this resonance, that a very, very low energies is created, this resonance that basically the systemic interconnection is one of the most important things. So you can think of the body as a sort of a container evaporating liquid, or you can think of the body as an alive mechanism that intercommunications. And aging is basically a systemic problem. It's not just the loss of one collagen of some, it's a systemic resonance problem. And this is me in our research. We've been doing research for 48 years now. I know you might say how old I am. I'm actually 71. And that's another shot from our research center. Resonance is what gets the system working together. Basically, you have the resonance is like the cable of the telepheric, and you have the stem cells is what that telepheric cable is carrying. So the resonance is the interconnectivity, is something that connects all the, and this is basically the main subject of my new book, The Rhythm of Youth, that has to do with interconnectivity, resonance, and systemic balance. And that's me now. This is my other books have been translated in many different languages. And with this, I would like to thank you very much. Thank you.